Hi, my name is Nirav Shah, and I want to thank the coordinators of this conference in Saudi Arabia for inviting me today to talk about bispecific CAR-T cell therapy. These are my disclosures. The reason for bispecific CAR-T cell therapy was really recognized early in the early days of giving CD19-based CAR-T cell therapy because one limitation that quickly became an issue with targeting CD19 alone was that selective pressure against a single antigen can lead to target antigen loss. And so in the initial clinical trials, such as Eliana, the majority of patients with B-cell ALL that relapsed actually had CD19 negative disease. Since then, in retrospective studies with diffuse large B-cell lymphoma, about a third of patients were found after CD19-based CAR to have loss of CD19, potentially being a mechanism of resistance to CD19 CAR T cell therapy. You can see here on the drawing on the right that there are multiple mechanisms that are sort of proposed, including sort of changes in the receptor genetic gene mutations within the receptor that allow it to no longer be expressed or to be truncated. Cell lineage switch is another way that tumor cells can downregulate uh, the CD19 receptor, and then there's been a case reported of epitope masking as well. So if CD19 is lost, do we lose our other B cell markers? So this was a really nice study done looking at patients who had CD19 based CAR T cell therapy. And what they found in these group of patients with non-Hodgkin lymphoma is that even though a subset of patients lost CD19 after CD19 directed CAR T cell therapy, the CD20 expression remained high and basically stable, regardless of what happened to CD19. And so this data sort of helps support the concept that dual targeting might be a way to mitigate at least one mechanism of resistance to CAR T cell therapy in its current form. So, what does the future of CAR T cell therapy look like? This is a nice drawing I found on the internet, and it goes to show all the different approaches that are currently under investigation to use cell-based therapies to target the tumor cells, from using allogeneic products to armoring the CAR to make them sort of more resistant to the immunosuppressive effects of the tumor cells. To what we're going to focus our conversation on, which is dual targeting or targeting of more than one receptor on the malignancy. So, in the world of B cell malignancies, there are sort of well known B cell receptors that we have been harnessing in terms of treating malignancies for many years now. Most commonly, actually, CD20 was the first major target with the drug called rituximab, which is the mainstay in the treatment of B cell non Hodgkin lymphoma. But we now have CD19 targeted agents in lymphoma and leukemia with blinatumomab, loncastuximab. And we also have a CD22 targeting agent with inotuzumab. And so these are the most common and prevalent targets. And as a result, most of the bispecific CARs that are being developed are either combinations of CD19 and CD22 or CD19 and CD20. This is not to say that new iterations of this won't develop as we identify newer and better targets on B cells, but for the time being, these are sort of the mainstay bispecific approaches that are being utilized. So this is an important slide that I want to sort of review in detail. There's more than one way to build a bispecific CAR. And so I want to sort of go through the different approaches that different groups across the country and the world are taking to do a dual targeted approach to cell therapy. So one approach is co-administration. In this setting, basically what you're doing is you're making an aliquot of CD19 targeted CARs and an aliquot of CD20 targeted CARs, and you co-administer them. The problem with this approach is that sometimes you can get differential engraftment with the CAR that tends to bind a little bit better. And so you might see variable levels of activity of these two different CAR products that may compete against each other. Another approach is to have a bicistronic CAR. So now you're using one sort of gene construct and you're modifying the T cells once, but you're modifying them to have more than one single chain variable fragment, allowing it to sort of bind different epitopes on the malignant cells. So you can have an anti-CD19 and an anti-CD20 built within the same CAR T cell. A third approach is something called co-transduction. Now, in this situation, you're not making two separate products, you're making one CAR T cell product. And what you're doing is you're adding lentiviral vectors that can actually code for different CARs. The problem with this approach is that you're going to get sort of a variable expression of different CAR products. You'll have some CARs that are just CD19 targeted, some CARs that are CD20 targeting, and some CARs that actually, the construct infected both, and so you're going to have CARs that have both the CD19 and CD20 being expressed. And then the last approach is to have a tandem receptor. Now, in this situation, you use a single sort of viral vector, but it codes for a construct that has two epitopes that are connected by a linker. And so you have one sort of CAR molecule, but it is able to bind two different epitopes and two different antigens on the target cell. So, I'm going to talk a little bit about what we've been doing at the Medical College of Wisconsin in Milwaukee in the United States. What the approach we have taken was to use a bispecific construct targeting CD20 and CD19, and we used a tandem CAR. And so, this is again one single chain variable fragment within, or one CAR molecule with two single chain variable fragments targeting both CD20 and CD19 connected by a linker. We did a phase one clinical trial, sort of a dose escalation study, evaluating this construct in patients with relapsed refractory B cell non Hodgkin lymphoma or CLL. We used standard flu-cy lymphodepletion, and we made the CAR T cells using the Prodigy Clinimax device, which allows for point of care manufacturing. And we used IL-2 for cell expansion and identified a dose of 2.5 million cells per kilo for further exploration. This goes to show sort of our response data. So how well do bispecific CARs work in a clinical trial setting? You can see here that the green bars represent patients who achieved a complete response. I've limited this to the patient population that got the full dose of CAR T, the 2.5 million, because we found the patients who received a dose less than that did not have durable remissions. You can see here, though, that there was a group of patients that got the full dose and also, because of our point of care manufacturing ability, got a fresh infusion and really had the best response rates. So these are patients where we did not need to cryopreserve the CAR T cells down. And that's something that you can only really do if you're making them in-house and delivering them in a point of care fashion. But what you can see here that the bispecific CAR is efficacious and does lead to durable responses. The bigger question with the bispecific CAR, though, is does it mitigate the mechanism of resistance that I mentioned earlier? So, can we sort of minimize CD19 negative relapse? And so, we did actually biopsy every patient. Again, this is a small sample size and not representative of what would happen in a larger population. But of the patients that did progress, and you can see here, about half of them were patients who did not receive the full dose of CAR T cells so received sort of a lower dose as part of dose escalation, we found that CD19 negative relapse did not occur in this patient population. And so, for the most part, CD19 expression was stable among the patients who relapsed, suggesting that mechanisms of resistance to a dual targeted or bispecific CAR may be different than what we see with the single targeting CD19 CAR T cell product. One thing that we did find is that we did have a subset of patients that had late relapse. And what late relapse means is so the way we defined it was relapse that occurred greater than 6 months after the CAR T cell treatment. And we actually found that the majority of those patients were those who had limited in vivo expansion. So the peak expansion in the first 28 days was predictive of long-term clinical outcomes. So our key takeaways from this initial study was that bispecific 2019 CAR T cells have a promising safety and efficacy signal at a dose of 2.5 million cells per kilogram. We did not find, at least for the majority of patients, that the relapse was due to changes in CD19 and CD20 expression. And we found that there was a group of patients that initially responded but had a late relapse, and that was due to poor in vivo expansion in the first 28 days. So the question our group asked next is there a way to optimize this bispecific construct? There seems to be some advantages in at least preventing CD19 targeted loss. And so we developed a new trial to sort of optimize this construct. What did we do? Well, we shortened the manufacturing length down. There's some data out there that by extending the manufacturing time, you can actually lead to a more terminally differentiated or exhausted CAR T cell product. And then we modulated the cytokines we use to produce the CAR T cells using IL-7 and IL-15 in lieu of IL-2. The idea here being that that would be a more robust cytokine population and could lead to a better CAR T cell product. This was sort of our clinical trial design. We had a safety run-in using this IL-7, IL-15 combination, and then open multiple arms, including in mantle cell lymphoma, and then to understand if length of manufacturing made a difference at sort of a 8-12 flexible process where we harvested at day 8 and a fixed 12-day process as well. So this is some data we presented at the TCT meeting for this ongoing study here. At that point, we had enrolled about 29 patients. You can see here that this is a patient population mostly with diffuse large B-cell lymphoma and mantle cell. The median age was 64 years. The median lines of therapy were 4. And the majority of patients, again, using our point of care approach, got fresh CAR T cells. Compared to IL-2, and I didn't share this data here, the combination of IL-7 and IL-15 improved the transduction efficiency and resulted in a higher number of CAR T cells in a shorter period of time. But overall, you can see here that the demographic population suggests that this is a very sort of relapse refractory group of patients that have seen multiple prior therapies. These were our clinical outcomes here. You can see a high overall response. Um, so these are safety outcomes here. You can see overall a favorable safety profile. There was one grade 3 CRS event in 29 patients, 3 patients with a grade 3 ICANS. We had one dose limiting toxicity in a patient that had a prolonged grade 3 ICANS and a grade 4 acute kidney injury. And our day 90 non-relapse mortality, we did have 2 patients who passed away from infectious complications. But for the most part, the toxicity was mild and manageable. And one thing that we did find is that the rates of CRS were higher in the 8-day manufacturing arm, again, suggesting that these CAR T cells might be a little bit more naive and more robust, leading to maybe higher CRS in that subset population. The majority of patients here did get a dose of tocilizumab, and you can see about half the patients received some degree of steroids for management of their toxicities. These were our clinical outcomes at day 28. 90% of patients had an overall response. So that was a CR or a PR. We did have 3 progressive disease patients at day 28, but I would note 2 of those 3 were CD19 CAR T cell failures. So patients who had prior exposure to a CD19 single targeted CAR and then progressed and then went on this clinical trial. So for our CAR naive, our response rate is even higher. And we were able to assess MRD negativity in a subset of patients and found that about 15 to 20 were MRD negative. Again, we're using the commercially available adaptive clonoSEQ assay. You can see here that the overall response rate looked a little bit better in the 8-day arm with the higher CR rate. Again, is how clinically meaningful is that is something we're still trying to determine. And you can see here that we had a mantle cell cohort with excellent outcomes and an overall response rate of 100%. One thing we wanted to note is we did look into whether the length of manufacturing impacts the CAR T cells, and we did actually find that our hypothesis was correct. The products that were harvested at 8 days of manufacturing did demonstrate a higher percentage of central memory and T stem cell memory phenotype T cells than those products that were harvested at day 8, where those cells nearly became absent. And again, the clinical significance of this is unclear, but the general thought is that the higher percentage of central memory or more naive T cells can lead to more durable and persistence of CAR T cell responses, and it may be a way to optimize the product. And hence, shortening manufacturing is a methodology being used across the CAR T cell field. So I highlighted some of the work that we've been doing at the Medical College of Wisconsin, but I really wanted to, inform people that the dual targeted CAR has really taken off in the entire world. There are multiple other people using dual targeting to address uh, the single targeted CAR T limitations. So this was actually a paper that came out of China. They had a TAN CAR 2019, so TAN standing for tandem CAR, and they actually looked at multiple different variations and built several different tan cars and ultimately found that the tan car 7 was the optimal construct that they selected for human use. And you can see here that their construct is actually very similar to what we have. It's a CD20 distal targeted with the linker, and then a CD19 targeted with a transmembrane molecule with CD8 and a similar 41BB CD3 zeta CAR. They treated 28 patients, 19 had diffuse large B-cell lymphoma or transformed follicular. And again, they saw very nice responses, an overall response rate of 79%, a CR rate of 71%, and a 1-year progression-free survival of 64%. They did not report any grade 3 ICANS, but did have a higher grade 3 CRS rate of 14% and an overall CRS rate of about 50%. Again, there's other products that are still developing. Kite, the company that makes Axicell, has Kite 363. This is an autologous CAR T cell product, and this uses a bicistronic vector. So again, it's going to be a CAR T cell with sort of two separate CAR molecules on it that are able to target both CD19, and this is combined with a CD28 co-stimulatory domain with a separate CD20 single chain variable fragment and a 41BB co-stimulatory domain. So this is going on in phase 1 clinical trials, and I think we're all excited to see how this does. There's another group by Dr. Yvonne Chen and UCLA. They reported some of their data at the TCT meeting and ASCO, and they also have again a similar. This is a tandem CAR, CD20, CD19. It's a 41BB, CD3 zeta, so similar to the TAN CAR and then the CAR that we have been developing at the medical college. And again, here they treated 8 patients, and so far 7 are in ongoing remission. So again, we're seeing high response rates. I think for these two products, really too early to know how different they are from the other dual targeted CARs that are being under development. I did mention that I focused so far because of the work that we're doing on CD19 and CD20 targeting, but as I mentioned, the other combination to consider is CD19 and CD22. This was a product that we heard about a couple years ago. Still waiting on additional updates here, but this was an anti-CD19, anti-CD22 bicistronic CAR T cell product, and they were studying it in combination actually with pembrolizumab. So the idea of using a PD-1 inhibitor to sort of maybe mitigate the immunosuppressive effects of the tumor microenvironment. And they treated 33 patients with aggressive B-cell lymphoma. And again, we see a nice response rate of 69% and a CR rate of 52%. And the majority of patients, at least with limited follow-up, who achieved a CR maintained that CR. However, they only had 3 months of follow-up time. They commented on no grade 3 to 4 CRS and 9% neurotoxicity in this patient population. Again, haven't heard too many updates about the progress of this, but looking forward to learning more about this combination. This was a very nice publication of a tandem CD19 CD22 CAR T cell product. This was developed by Crystal Mackel and the team at Stanford University. And so they used this construct actually in two different cohorts of patients. They used it in B cell non Hodgkin lymphoma and B cell acute lymphoblastic leukemia. And so in the non-Hodgkin lymphoma group, they had an overall response rate of 62% with a 29% CR rate. You can see that their long-term progression-free survival is about 25% of patients. But interestingly, they did, unlike the 2019 studies where we haven't seen that much, they did find that CD19 negative relapse occurred in 4 out of 14 patients. In B cell ALL, they had a very nice overall response rate in MRD negativity. In general, this is consistent with just sort of CAR T cells in general for ALL. We see high response rates. The problem is that people continue to relapse, and so you can see here their progression-free survival on the right as well. That the majority of patients ultimately do end up relapsing or passing away, and they again did find that CD19 negative or CD19 low relapse did occur in about half of the patients who had relapsed. And so, what does this go to show? It's that while CD19 and dual targeting may be a way to mitigate some degree of CD19 loss, but a conclusion here is that CD19 antigen does remain a mechanism of resistance even after a bispecific CAR T cell therapy. This is another CD2219. This is again from a group in China. And this is a small sort of phase 1 trial looking at different cell doses. And again, using a similar tandem CD19, CD22 construct. and they found high degrees of response rates. All patients, 6 patients received an MRD negative remission, but at the time with limited follow-up, one patient did relapse, and again, they found that the patient did escape with CD19 negative and CD22 dim disease. Overall, the toxicity of this was in line with other CAR T cell therapies in ALL. Lastly, there's a group sort of doing the co-administration methodology. So this idea is you make two different aliquots of CAR T cells and you sort of administer them in a co-administration fashion. So you can see here, this was a trial coming out of the University of Pennsylvania by Dr. Fry. And so they gave lymphodepletion and they sort of did this stepwise dosing and they gave 10% of CD19 CARs and CD22 CARs on day 0 and they fractionated the dosing, holding subsequent dosing if they had early signs of cytokine release syndrome. And so again, a very, very high response rates, 11 of 13 patients responded. And with the median follow-up of 10 months, there was only one molecular relapse. And so again, you can see here that everyone has the same concept here that we need to hit the tumor cell with more than one target, ideally to minimize relapse, which remains to be a barrier with CAR T cell therapy. And then lastly, what's the future of all of this? Well, if two targets are good, why not three? So there's a group, Dr. Schneider, that published this report using a tri-specific or multi-targeted approach. And so this sort of harnesses multiple technologies in order to build a construct like this. They called it a duo CAR, but really it's a bicistronic CAR where one of the constructs is actually a tandem. And so their tandem is a 2019, and then they have a bicistronic CAR sort of construct with a single chain variable fragment that targets a CD22. And so this is a way of actually hitting, multiple B cell markers, probably, the idea here being leading to a very deep B cell ablation. We are actually working with Dr. Schneider's group, and we launched a phase 1 clinical trial using this triple construct and have just enrolled our first patient in a dose escalation study. Don't really have any updates here to tell you, but we're looking forward to sharing them as we continue to treat patients with this new novel construct and really trying to learn if multi-targeting of B cell receptors can actually overcome some of these resistance patterns we're seeing in terms of B cells escaping by downregulating CD19, CD22, or CD20. Just to know that we're not the only ones doing bispecific CARs. In B cell malignancies, this is happening in multiple myeloma. There was a nice report at ASH of using a CD38 BCMA bispecific CAR T cell. This had an overall response rate of 87%, a median progression-free survival of a year and a half. There's also these CARs under development using a BCMA GPR C5D CAR T cell construct. So again, trying to target the multiple myeloma cell in more than one fashion. In diseases like AML, actually multi-targeting is really a favorite approach because there's a lot of heterogeneity and they don't have nice markers like CD19 in B cell malignancies. So there's multi-targeted approaches targeting CLL1, CD33, and CD123. And then there are other novel combinations in B cell lymphoma as well, using things like BAFF and CD137 for dual targeting. So to conclude this talk, I talked a lot about all the different ways we can do dual targeting. I talked about, the work that we have been doing at the medical college and some of the publications of bispecific or dual targeting of B cells that are really happening across the world. But, despite the excitement of dual targeting, what I really wanted to note is that at this time, it really is unclear if bispecific CARs actually, decrease the number of patients that are going to have downregulation of target receptors. And more so, everything I've shown you are single arm studies in a highly selected patient population. So these are phase 1 clinical trials. They have strict eligibility criteria, high patient selection. So the biggest question is does dual targeting actually improve overall response rate? Does it improve duration of response? Does it improve progression-free survival? And we need larger data sets with a more homogeneous population, maybe patients that are more real world than what we presented to actually answer that question adequately. But what I can say is that there is a lot of excitement. I think the one limitation of dual targeting is that it doesn't really mitigate other resistant factors. So we've learned, and there's been nice publications that show that the tumor microenvironment, T cell exhaustion, recently the presence of Tregs in the CAR T starting population, that all of these things can also impact the ability of the CAR T cells to be effective. And so just increasing the number of target receptors may not be enough to actually improve the response rate with CAR T cells. Again, all of this is really to be determined, but so far I think there is very exciting science trying to improve on the current standard of care of CD19 targeted CAR T cell itself. I think I want to sort of conclude here. I hope this was a quick overview, helping you understand bispecific CARs, how they're built, what the different approaches are, and sort of what the state of the science is for these products right now. I'd like to thank actually for the research that I talked about at the medical college, an incredible group of scientists and collaborators that have helped us put this work together, and of course the patients that have participated not only in our clinical trial, but all of these clinical trials to help advance science. With that, I'll be joining the Q&A session, and I thank you all for your time today. Hope you enjoy the rest of the meeting.